Today I will present on the paper combining machine learning with a pharmaceutical technology roadmap to analyze technological innovation opportunities. Contents are followed and I start with the introduction. New drug development faces challenge due to high costs, long development cycles, and low success rates. And existing TRM failed to consider clinical trial basis and primarily focus on analyzing papers and patents. Also, expert-dependent evaluation methods lack objectivity. So this research objective is that to integrate TRMs with machine learning to automate the analysis of pharmaceutical technology innovation opportunities. Also, to overcome the limitation of existing TRMs, a six-layer trial model is developed, covering technology, clinical trials, products, market categories, and future direction. Paper patents and commercial data are comprehensively analyzing using BERT's and LD model. And next one is theoretical background. Previous studies have used quality methods such as Delphi model, and this approach is time-consuming and reduces the reproducibility. To address this limitation, bibliometric analysis have been introduced. However, it has also problems. So, to address this, text mining has been introduced to explore pharmaceutical technology innovation opportunities and commercial applications. Recent studies increasingly adopt ML approach to automated pharmaceutical technology innovation opportunity analysis. ML-based analysis integrate research papers, patents, and clinical data to predict technological evolution paths and commercial feasibility. TRM is used to visualize the development trajectory of pharmaceutical technology innovation and analyze future technological trends. Traditional TRMs follow a three-stage structure: technology, product, market. However, in the pharmaceutical industry, clinical trials play a crucial role to necessitated an improved TRM framework. For drug innovation, clinical trials account a lot of amounts of R&D cost. Recent studies integrate TRM with text mining and machine learning techniques to enable more precise innovation opportunity analysis. ML-based TRMs allows for real-time analysis of relationships between patents, research paper, and commercial data. The integration of TRM and ML enables a more structured and quantitative analysis of technological evaluation. ML techniques have been increasingly used to support quantitative and intelligent TRM analysis. The main tasks of ML include classification and topic clustering. Some studies have applied ML and text classification models to automated layer-wise classification with TRM. Other studies have combined ML with topic clustering models, and one of the most widely used ML model is BERT. Google introduced the BERT model in 2018, which significantly improves ML classification accuracy. However, pre-trained BERT models do not perform optimally in domain-specific tasks, requiring fine-tuning with domain-specialized dataset. To address this study develops a small domain-specific dataset and applies a fine-tuned BERT model for pharmaceutical data classification. Let's look framework overview. At Module 1, data is collected from Medline, DII, and ABI. At Module 2, a BERT-based classifier automatically categorizes data into TRM layers such as technology, clinical trials, products, and market. LD modeling clusters key technological topics and organize them within TRM layers. At Module 3, cosine similarity analysis tracks the relationships between technological topics and their evolution. This study collects data from three databases: Medline, DII, and ABI. Each database serves a different purpose. Medline contains biomedical literature, DII provides pharmaceutical patent information, and ABI includes industry and market data. To retrieve relevant data, scientific abstracts are such as using medical subject headings terms while patent data are filtered through the IPC code. Now, we will go over the technology roadmap design in this study. This TRM is designed specifically to analyze pharmaceutical technology innovation opportunities. The vertical axis of the TRM consists of 6 layers. The first layers is technology, represents drug research and development capabilities in achievement. The second layer is clinical trial, includes phase 1 to phase 4 clinical trials, and third layers is product. Focuses on product layout and generation. The fourth layer is markets, covers sales and market share. The fifth layer is categories, classifies key technologies. Finally, the sixth layer is future direction, outlines the future developments paths of pharmaceutical technologies. The time axis of the TRM is divided into 4 stages based on the technology life cycle. These stages are emerging growth, maturity, and saturation, which help on tracking how technologies evolve over time. A BERT classifier is applied to automatically categorize data into 4 TRM layers: technology, clinical trials, products, and market. Once the data is categorized, NLP techniques are used to refine the data by removing irrelevant contents and grouping semantically similar data. Finally, domain experts curate the training data to fine-tune the BERT model, further improving classification accuracy. The classified data from the previous step is divided into n by n subsets, each corresponding to a specific TRM layer and time stage. The preprocessing step includes word segmentation, verb reduction, manual combination of similar words. Also, TF-IDF method is applied to remove overly frequent or rare words. A stop word list for domain-specific terms is created with the help of experts. LD model is applied to automatically cluster key pharmaceutical technology topics. Perplexity scores are used to determine the optimal numbers of topics. The alpha and beta hyperparameters in the LD model are optimized to improve clustering. The LDA space package is used to visualize the cluster topics, where circle size indicates the strength of technological activities and the distance between circles reflects topic differentiation. Cosine similarity is applied to measure the similarity between topic clusters using LDA. Also, a threshold value is set to determine whether a potential connection exists between topics when the similarity exceeds their threshold. This allows for analysis of how pharmaceutical technology topics are interlinked and evolve within the TRM. The first step to draw pharmaceutical TRM is visualizing the TRM using LDA basis. The second step involves identifying potential links between topics within the same or different layers. Topics with similarity values existing threshold value are connected within the TRM with different colors representing different categories. Based on the results, technical topics are classified into 4 categories according to the continuity and strength: key topics, hot topics, breakthrough topics, and blank topics. Finally, major pharmaceutical technologies topics from L1 to L4 are integrated to define key technology categories in L5. Also summarize future development direction in L6. Now I applied this method to hyperuricemia drug. This study collects data from 3 databases, and by using medical subject heading terms such as goats or hyperuricemia, the research team retrieved papers from Medline. Patent data were collected using the IPC code, and commercial data published in ABI was retrieved using the keyword hyperuricemia. Let's look at TRM design. The TRM is designed as a framework to visually analyze pharmaceutical technology development. The time axis of the TRM is divided into three phases: introduction, growth, and maturity, to track the progress of technology development. After integrating paper, patents, and commercial data into a single corpus, the data is classified into S1, S2, and S3 based on technology development stages. This allows for an analysis of how pharmaceutical technologies evolves and its impacts on the market. Extracts tech-related data features from papers and abstract in S1, S2, and S3. Based on Google's pre-trained BERT model, three domain experts specializing in pharmaceutical innovation and hyperuricemia drug development constructed a domain-specific training sets. To improve performance, the BERT model was fine-tuned and each subset was classified into five groups: L1, L2, L3, L4, and L0. Only L1 to L4 were retrained per stage, and 3,000 sentences were randomly selected as a validation set. Finally, the 76,793 short sentences were divided into 12 subsets, mapping their positions within the TRM. To the next, pre-processing data, numbers and basic verbs are removed and stemming and limitations are performed. Domain-specific stop words such as hyperuricemia and therapeutic are added and removed from the analysis. The TF-IDF method is applied to eliminate overly frequent and rare words. To cluster and extract the technical topics of hyperuria disease based on LDA. The hyperparameter of the LDA model are set to alpha to 50 per K and beta to 0.01. The optimal numbers of topics for each dataset subset is determined based on the perplexity values ranging from 2 to 50. Each topic is described using 30 related words with the top 5 keywords listed. The result of the LD model cluster key pharmaceutical technology topics across to 12 sub-datasets. And these are table 4 to 5 and figure 2. Let's more talk about the figure 2. The first step in TRM framework, structuring them across L1, L2, L3, and L4, and aligning them with time stages S1, S2, and S3. In the second step, topics with cosine similarity values of half or greater within the same sublayers are linked, with different arrow colors representing connections between related topics. A connection between different layers are established if the similarity value is 0.35 or greater. Key topics are marked with red triangle, hot topics are marked with green circles, and breakthrough topics are marked with quadrilaterals yellows, and blank topics, rose-red pentagons, were identified. With expert input, the studies categorized hyperuricemia drugs into three key types: Chinese medicine, small molecule drugs, and protein drugs. And the following part is an interpretation of the results, so it is not important for us, so I will skip it. Let's look conclusion. This study proposed a systemic methodology for designing pharmaceutical technology roadmap and analyzing technological topics. This study expands on the traditional market product technology TRM structures to market, product, clinical trial, and technology TRMs, forming a domain-specific. This study develops a data-driven method for identifying technological opportunities using BERTs and LDA. This approach effectively tracks the evolutions of the technological topics and provides a novel framework for analyzing pharmaceutical innovation opportunities. This study primarily used research papers, patents, and commercial data as data sources for the TRM, but didn't include additional product data such as pharmaceutical databases. Although the study utilized machine learning tools such as BERT and LDA to construct the TRM, domain expert involvement was still required in building the small training datasets. Thank you for listening.